It's my great pleasure to introduce today's speaker, Dr. Allan Butterfield from University of Kentucky. Allan received his bachelor degree from University of Maine and his PhD degree in physical chemistry from Duke University in 1974. He has been a faculty member at the University of Kentucky since 1975. He's currently the alumni associate endowed professor of biological chemistry and director of University of Kentucky Center of Membrane Science. He has many other titles as well, which I list out first slide. So Dr. Butterfield's research interests across many disciplines, and his research on biochemistry of beta amyloid provides significant novel insights about how beta amyloid drives Alzheimer's disease pathogenesis through increased oxidative stress. He's a very productive scientist. He's published over 554 papers. A number of them are highly cited. And he received numerous awards and honors, which the list is too long for me to read here. But I would like to point out one. He was a recipient of the President's Award for Excellence in Mentoring Female Scientists, which was presented to him by President Clinton in the White House in 1998. So as a parent of two girls who are really interested in math and science, I appreciate very much his dedications to training women scientists in this. And today, the title of his talk is Redox Proteomics and Alzheimer's Disease Insights into Neurodegeneration and Progression of his dementing disorders. Okay, welcome.
Thank you. Thank you very much. So it's a real pleasure and an honor for me to be here, a privilege really, to share with you some of the research done by my research group. And I've really enjoyed myself today and met a lot of very good people and good scientists. So let's start with a definition. What is free radical oxidative stress? So basically, it's a toxic condition in which there's too many free radicals being produced or a failure of antioxidant defense systems, or often both happen. And of course, there's a wide list of oxidative stress, ROS and RNS. And brain is particularly poised for oxidative damage because it, even though it's only 1,200 grams, it consumes a third of the inspired oxygen in your body. It's rich in polyunsaturated fatty acids, which are perfect targets for free radical damage. And it's rich in transition redox metal ions like iron and copper, which facilitate free radical reactions. But our brains, unlike other organs, is relatively low in antioxidant capacity, so it's primed for free radical damage. So when a free radical is formed, for example, in the lipid bilayer, then either the free radical, or more likely the free radical reaction product, for example, HNE, which I'll speak about, have immediate protein targets to attack. And as soon as that happens, it will bind to these proteins at various amino acids and induce a conformational change within the protein. And every chemist knows, and I bet every of you have been through chemistry at one time or another, function follows structure, not the other way around. And so when the structure changes, the function changes. And so that is really important to know. So, and usually detrimentally. So one of the ways of looking at oxidation of proteins is by making the hydrazone between DNPH and the carbonyl. The carbonyls always show up on proteins when they're oxidized. This was first pioneered by Earl Stadman many years ago, a great friend of mine, before he passed away. And when you make the hydrazone, that thing can be easily detected by immunochemical means and quantified. And for lipid peroxidation, our lab uses the formation of 4-hydroxy-2-noninal, which is HNE. So HNE can covalently bind to cis, his, and lys. And if you remember your organic chemistry, it's a process called Michael addition to make a covalent adduct here. And another one of these is acrolein, which has the same chemistry. And then the third marker we use for oxidative stress is 3-nitrotyrosine. And so nitric oxide and superoxide are two relatively not so reactive free radicals. But in the presence of each other, radical-radical recombination is about the fastest reaction known. And so it will form peroxynitrite, and in the presence of CO2 makes this nitrosyl peroxyl carbonate, which rearranged into nitrocarbonate, and that homolyzes to give to you the NO2 dot radical. And then in the presence of tyrosine in an oxidative stress environment, like an Alzheimer's brain, that phenolic OH is going to be lost. And because OH is orthodirecting substitution, or substituent on a benzene ring, then it's the 3 position in which that will delocalize. And now you get, again, radical-radical recombination and get the 3-nitrotyrosine formed here. So with that background, think about this. This is a Newsweek article from 1984. Nobody really was given a whole lot of thought to Alzheimer's disease then, but they sure are now. There are 5.5 million Americans currently with Alzheimer's disease, and because of people like me and some of you who were born after World War II and before 1963, there are 80 million of us. I was born right after World War II, and there are 80 million of us baby boomers. Aging is the single most important risk factor for the disease, and so it's predicted, depending on who you listen to, somewhere between 15 and 20 million Americans will have Alzheimer's disease in 30 or so years if something isn't done to slow down the rate of progression of this disease. And so Alzheimer's disease in this department and this university, especially with the Barshop Institute on Aging, is so well known as it is, and many other departments. I won't spend a lot of time on this. So if you have more questions about AD, let me know and we'll talk about it. But I thought you might be interested in this. This is so we believe, is the world's fastest autopsy program. We always get samples within 2 to 4 hours of death, and never more than 4. And so we have about the fastest time you can get, as close to a living brain as possible. So here is a slice through the brain of an Alzheimer's patient, and just look how large the foci are. I mean, those should be filled in with neurons, and they're gone. So no wonder higher executive thinking and cognitive function don't take place. And look down here. I mean, basically, the hippocampus is gone. So how we could possibly make memories if you don't have that structure present? So in a pathological hallmark of AD are these senile plaques that have this aggregated damaged peptide that's called amyloid beta peptide. And many people believe that A beta is central to the pathogenesis of AD. And that's mostly brought about by considerations of genetic concerns. If somebody has a mutation in any one of these 3 genes, 2 things always happen. The person always gets Alzheimer's, and there's always a huge deposition of A beta 42 in the brain. Everyone with Down syndrome, if they live long enough, will get Alzheimer's disease. And so, and what's so interesting about that is the trisomy of chromosome 21 is where APP, from where A beta comes from, is also coded. So you get a dose response, a dose effect here, and so you get more A beta being produced on this. Now, in our lab and Bill Marksbury's lab for the DNA and RNA oxidation, his lab did that. But for lipid and protein oxidation, I think our lab is probably the first or among the first to measure the free radical oxidative damage in Alzheimer's disease and mild cognitive impairment. And we see, using different kinds of markers for lipid peroxidation and protein oxidation, and Bill had done DNA and RNA oxidation. So we tried to combine the idea of these 2 things I've just told you. A beta is central to the pathogenesis of AD on the one hand, and the Alzheimer's brain is under oxidative stress on the other, into a single model. And the model is sort of like this, where A beta, as a small oligomer, inserts into the neuronal lipid bilayer, undergoes lipid peroxidation, produces HNE, and attacks key transport proteins. And among these are the ion-motive ATPases. So with the sodium potassium ATPase and the calcium ATPase damaged, then all of these things I'm saying to you we have shown to be the case, by the way, then the voltage, the cell potential changes. So voltage-gated calcium channels open. In comes calcium. Students, how many students are in here as opposed to postdocs? Any students? Do you know the difference in calcium concentration outside a neuron versus inside? It's about 10 to the fourth, right? So the chemical potential of that concentration gradient will drive calcium into the cell. And when it comes into the cell, I will give you a little more detail later, but basically, as they say in Kentucky, all hell breaks loose. And so what happens, there's no chance in the world that neuron is going to survive because we have endonucleases, proteases, ER stress, mitochondrial swelling and death. And so that neuron is going to die. When enough neurons have died, you see the symptoms appear. So given that, we tested this model in vitro, for example. Now, this is just a summary of many years' worth of work in the lab to show that we could detect ROS and cell death in neuronal cultures, and vitamin E could block all that, which is what, or elevate glutathione could block that. So this is what you might expect then of a free radical reaction. And protein oxidation was elevated, and again blocked by these antioxidants, and lipid peroxidation was elevated, blocked by these things. So this is exactly what you'd expect in a free radical reaction. And then these data are from Mark Mattson, who used to be a faculty member at UK, and then now he's head of the National Institute on Aging internal intramural research in Baltimore. And what he showed was if after I think we demonstrated in 1994 the A beta causes lipid oxidation. This paper came out in 1997. He took he took cell cultures and treated them with amyloid beta peptide, and then look at the soup above the neurons, and he could measure HNE in the control cells and see a little peak. And then after A beta, you see a huge peak. So it's absolutely clear that HNE is being formed. And that concentration was in the micromolar rate. So it's a huge bolus of HNE that's being formed. One more thing that I don't have on the slide, but you should appreciate. Lipid peroxidation cannot occur, absolutely cannot occur, unless the free radical that initiates it is in the bilayer. Cannot occur from a radical outside the bilayer because they're too reactive and will never get to allylic hydrogens to initiate that first step. So that means the A beta has to be in the bilayer in order for this to happen. That's consistent with our model. So here is this is the cover of the most recent ARS that is it's our figure we made of a hypothetical protein with HNE on it, and you can see it bound to cysteine here. Here's the carbonyl. And so what happens is when this binds, it changes the conformation of the protein, and we demonstrated that. Well, I guess you can't see that over there, but we demonstrated that first in 1997 using spin labeling to show when HNE is added to a protein, the conformation of the protein changes. This was in synaptic membranes in that case. So with that idea, we tested some years ago a protein called the glutamate transporter. And this is GLT1, or sometimes it's called EAAT2. And what you see in Alzheimer's is about a 60% elevation of HNE bound to this transporter compared to control. Now, Mazzalia at San Diego had shown earlier that this transporter didn't work, and now we can tell Eliezer why it doesn't work. Because this protein now is a different conformation as a result of oxidative modification. Now, in this left side, we repeated this experiment, this time in a preparation containing the transporter, and the only thing added new is amyloid beta peptide. And what you see is you get exactly what you see in the AD brain brought about by the peptide. So the peptide is able to induce the HNE in the bilayer of the synaptic preps, synaptic membrane preps, and thereby inactivate the transporter. So given, now I didn't have time to talk about that we'd earlier demonstrated that glutamine synthetase is damaged and aggregate and dysfunctional in the Alzheimer's brain. So there are two ways of getting rid of extraneuronal glutamate that don't work in AD. And so you increase the risk then of excitotoxicity, which certainly could disrupt calcium homeostasis and lead to cell death. Now here's another idea. You know, this is some work that's supported by an R01 we have, is you could have excess A beta in the brain of an Alzheimer's patient either by one of two ways, at least two ways, there might be more. But at least two ways is it's made more or cleared less. And the major efflux protein from the brain to the blood of A beta is LRP1, the same one that gets rid of ApoE, for example, from the brain to the blood. And what we used is the same idea that we used for the glutamate transporter a couple of years ago and showed, sure enough, that transporter also has more HNE on it and therefore is likely to be dysfunctional, and that would make sense for this idea. Now, there are some other proteins I haven't had a chance to, time-wise, to tell you about. MRP1, for example, we showed many years, 2004, MRP1 has HNE all over it. GST, that is involved in making the glutathione conjugate to like HNE, and MRP1 pumps that out of the neuron. None of that works because these proteins are damaged because of this. So that's what I said there. Now here's another summary slide of a lot of work that we did about in vivo studies of A beta. For example, if you express human A beta in C. elegans, you see protein oxidation at a time before the deposition of the peptide. That's consistent with it. We used the Pittsburgh compound to show the plaque in the worm, and what you can see is you could get this oxidation exactly when the phenotype appeared, which was paralysis of the worm. This is a muscle promoter that carried the DNA of the protein, so it would go in the muscle wall of the worm, and the worm became paralyzed exactly when the protein oxidation was elevated, but before the deposition of A beta. So that's consistent with the idea of the oligomers. Yeah.
So when you did these experiments, one is that you measured that whether iron homeostasis or copper homeostasis is changed?
No. This was simply measure of protein carbonyls and that sort of thing. Okay. And then we used this Pittsburgh dye to check for A beta. And then if we take human A beta 42 and inject it in the rat forebrain, in the nucleus basalis of Meinert, those are cholinergic neurons, and they project to the outer molecular layer of the hippocampus. So we measured in the hippocampus and saw a huge amount of increased oxidative damage. And Earl Stadman had long ago shown that HNE, when it was formed, could diffuse away from its site of formation. And we're suggesting, well, maybe it's being brought somehow up to the hippocampus and it can damage the heck out of that. But that would be consistent with a known cholinergic deficit in AD as well. Then, if we look at brain of APP, PS1 knock-in mice, so these are not knockout, so they have the right amount of APP and the right amount of PS1. It's just that it's the human mutant form, but it has the mouse promoter, so it goes to the right place and the right amount. And what we showed is that oxidative stress correlates with how much soluble A beta you could find and prior to the deposition of the protein, so of the peptide. And then recently we did a paper where if you raise these mice on and you put NAC, an N-acetylcysteine, which is a glutathione precursor, in the drinking water, then the glutathione level in the brain goes up about 40%. And what you see is that at the end of 5 months, prior to the deposition of the peptide, we blocked all the oxidative stress. Now, if we take our 5-month period to correspond to the period in which A beta deposits, we also get a protection against oxidative stress, but not as robust. So the idea, if we translate this result to, say, humans, It would suggest earlier in the disease as possible, you could treat the patient against free radical damage, the more likely you are to be successful. And that's probably why almost every clinical trial against antioxidants that failed in AD is they start so darn late. The neurons are already gone. You're not going to rescue them if they're gone. And I'm going to talk to you, I hope, about beagles, but if I don't, just ask me about the beagle study, because beagles produce human A beta. They produce A beta the exact same sequence as humans. And so you can learn a lot from a beagle more than a rat. Okay, and I hope to talk about some work that Veronica helped us with, too. So now, if we want to know which proteins are oxidized, we are going to we're going to have to develop a method. So we invented this method called Redox proteomics, and to identify which proteins are oxidized in the brain in subjects with AD and MCI. And so if we're going to use, and so earlier on, so Bill Marksbury and Kenny Hensley, working with us, Kenny was my graduate student, showed that oxidative stress occurred in Alzheimer's brain. But what about amnestic MCI? So we are among the earliest in that. And so we see protein carbonyls up in hippocampus. We see HNE bound to proteins in the inferior parietal and hippocampal regions. And here is 3-nitrotyrosine levels. So that means MCI has oxidative stress. This is early in disease. There is no dementia here. These people have normal activities of daily living, but they're amnestic. They don't have good memory because that's part of the disease progress. But it's before dementia has occurred. And so this is arguably one of the earliest, if not the earliest, form of AD. And so we're learning a lot by comparing these. Now, I stole this slide from Dennis Selko's article many years ago in Scientific American, but it's just to show that the plaque content is not uniform in the Alzheimer's brain. Here's hippocampus, but look at cerebellum, basically is free of pathology in essence. So Kenny Hensley showed that in 1995, he showed where A beta is, you get protein oxidation, but where A beta isn't, you don't. So cerebellum is not more oxidized than control in AD. But hippocampus and inferior parietal lobule, which are always collected for us at autopsy, are. So how do we get at this? And the answer is Redox proteomics to look at which proteins are oxidized. So before I talk to you about Redox proteomics, I just want to give a shout out to all the molecular biologists in the room who have really helped us understand modern neuroscience. I mean, these folks have really done a great service to us in making sure we understand what's happening. But it's probably fair to say genes don't do the work of the cell. Proteins do. And so it's really important to know more than gene analysis. You've got to know about proteins. And that's where proteomics comes in. And in our lab, we have three avenues of our proteomic studies. We try to learn about mechanisms of disease. We try to identify disease-associated biomarkers. And we hope to select targets for specific therapy, all related to Alzheimer's, what I'm talking about today. So in 2006, we wrote a sort of definitive book, I think. We edited a book on Redox proteomics. It's about 1,000 pages long. So it's just about everything you ever wanted to know about Redox proteomics, and it's in there. So anyway, our lab's the first to use this in Alzheimer's. And it started with my first Italian student who didn't speak a word of English when she came to UK, but learned and graduated. Now is associate professor in the University of Bari in Italy. So any of these papers I'm glad to send you. And I commend to you these papers right here, these most recent ones in antioxidant redox signaling. This is a 45-page review, invited comprehensive review on Redox proteomics. So if you would like, I mean, it's an enormous review. So if you would like to know about this, I commend that to you. So how do we go about it? So we have our set tissue, like brain, and we prepare it and run a impregnated pH gradient strip with the samples from pH 3 to 10, or if we want to go narrower, we can go 4 to 5 or 3 to 4, or whatever the pH gradient is to spread out the proteins. And by running a current through that strip, it will separate the proteins based on their isoelectric point. And then you add to that, you add that strip to a regular 2D gel and make a two-dimensional Western blot as well, and stain one with cyparubi and the other you stain for antibodies against HNE or 3NT or protein carbonyls. And by a very sophisticated image analysis that uses PDQuest from Biorad, it does a spot-by-spot comparison between the control and the subject and determines the spot of interest, which is more oxidized in the blot. And so you measure the intensity of the blot divided by the intensity of the gel, gives a specific oxidation. And that protein you then cut out and do it in gel digestion with trypsin, and then use zip-tip cleanup and so forth, and then submit those peptides to mass spectrometry to do MSMS. And then based on the spectra, and what you do basically is you sequence each of the peptides, and then you send those sequences to a database, and you know immediately what the protein is because you've sequenced the peptides. And then we have, every time we try something new, we always validate our findings to make sure there's no artifact in what we're doing. So those are the methods we use. And in Alzheimer's disease, these are the proteins, but don't concentrate on those yet, because I'll show them by function in a second. What I really wanted to point out is the bottom thing. When we repeat this using AD cerebellum, we cannot find one protein that's more oxidized than the control. It's exactly consistent with the pathology, because we don't see any A beta accumulation to speak of in the cerebellum. So if we look at these proteins by function, we see different things. For example, energy dysfunction. And I really want to point out that what we're looking for here are proteins that help us to explain three things: the pathology, the clinical presentation of the disease, and the biochemistry that you see. And I believe these proteins are all consistent with those, one or more of those things. So, for example, in energy dysfunction, if you look at the PET scanning, here's a normal brain, here's an MCI brain, and you can see already there's a diminution of glucose utilization. And look at an Alzheimer's brain, my goodness, it's really not using glucose much at all. And I argue that these proteins here are at least involved to some extent in that idea. So what I'd like to do now is just take a few examples of this list. We don't have time to go over the whole list, but just a few examples to illustrate what I was trying to get at. And the first one here is enolase that I'd like you to think about. So here's enolase over here in the glycolytic cycle. It converts 2-phosphoglycerate into phosphoenol pyruvate on the way to make pyruvate and enter the TCA cycle to make more ATP. So because we've also measured the activity, oh yeah, forgot one other thing to show you here. See these stars up here? That means we have measured, or the literature has measured, the activity of these identified proteins, and if it has a star, that means the activity is decreased, which is often the case. Stadman showed way long ago when proteins get oxidized, hydrophobic residues get exposed to the surface, and they tend to aggregate, and so therefore the active site gets messed up, and you can't, the activity goes down. So when these proteins are oxidized, they become dysfunctional. So there's enolase. We measured the activity, and sure enough, it's down in AD and probably contributes to what happens. So what happens if you have less ATP? Then you have a loss of cell potential, voltage-gated calcium channels open. You see calpain activation, caspase activation, endonuclease and phospholipase activation. And A-beta is able to replicate all of that. You can see MPTP opening, and so calcium ion accumulation in mitochondria. and cytochrome C coming out, apoptotic processes happening, and so forth. There's a loss of, when ATP is low, neurotransmission efficiency decreases. Phospholipid asymmetry. This is something I was talking about to the students and postdocs today at lunch. When phosphatidylserine comes to the outside of the bilayer, there's a protein called flipase. It uses ATP to flip it back in. But flipase has a central cysteine, one, that really regulates the whole protein. And it's a target for H&E. And so when it gets oxidized, it can't flip the PS. Now the proper term is PTDSER. When it can't get the phosphatidylserine back in, then as exposed on the outside, it's a signal for apoptotic processes again. Good example is in sickle cell. You'll see that pretty well, but also in brain. And then when ATP is diminished, you have excitotoxic processes, as I showed you earlier. But here's the story about enolase. Enolase is far more than a glycolytic enzyme, far more. It's a pleiotropic enzyme. It is involved in not only in the production of ATP, but it binds to membranes and stimulates pro-survival pathways involving ERK1 and 2. And it helps regulate the activity of plasminogen, which produces plasmin, which is a protease that cleaves A-beta-42. Both of those processes would be highly compromised with an oxidized and dysfunctional enolase. So that means enolase is really detrimental to neurons. And we'll keep coming back to enolase, as you'll see. Now here's GAPDH, standard old GAPDH. A lot of people use GAPDH as a loading control on gels. I'm going to suggest to you might think about that based on what we're doing here. So normally, GAPDH has many, many functions. So GAPDH interacts with SIRT1, which leads to the inhibition of P53, and that's really good for the neuron. Because GAPDH has an enormous number of cysteines, just an enormous number of cysteines in the tetramer, then it can react with glutathione and be S-glutathionylated. Well, that depletes the glutathione load, and that stimulates the pentose phosphate shunt to make more NADPH, and that helps glutathione reductase convert oxidized glutathione to reduce, and so it's all healthy for the cell. But on the other hand, those cysteines can also be nitrosylated. And when that happens, all of a sudden glycolysis goes to hell in a handbasket, and there's ion pump inhibition by the mechanisms I showed you, and calcium excitotoxicity and cell death. And also the VDAC1 can open, and cytochrome C and apoptosis-inducing factor can come out of mitochondria and lead to that. GAPDH is subject, because of the high cysteine load, to a lot of ROS and RNS, and that can lead to inhibiting of SIRT1, so P53 is left unchecked, and you can get apoptosis that way. And it is also known to be involved in APP and A-beta aggregation and in NFT paired helical filament formation. So there are lots of processes by which GAPDH, because it's oxidized, doesn't work that all are consistent with what you see in Alzheimer's brain. Now, I mentioned excitotoxicity earlier, so let's talk about the proteasome dysfunction. I know there are people at this institution who are very interested in the proteasome, and so let's talk about UCHL1, which is an identified protein. So when one has a damaged aggregated protein, then various ubiquitin ligases put polyubiquitin residues onto this damaged protein as a signal to move this to the 26S proteasome. And now, just before the protein can enter the proteasome, the ubiquitin carboxy terminal hydrolase L1 has got to take those ubiquitin residues off one at a time from the carboxy terminal side. A, there's a fixed pool of ubiquitin, so it's got to get recycled. And B, that's the only way this protein is going to fit into the proteasome for degradation and recycling. Well, that's the normal plan. But what happens in Alzheimer's disease is this protein is damaged and dysfunctional, presumably, and so it doesn't take those off. And now you're left with these ubiquitin chains on the damaged protein, which then can't be processed by the proteasome. So what do you predict? You predict things that are known in AD, that there are proteins with excess ubiquitin on them. And also, because of that, this is going to clog up the proteasome. There's going to be decreased activity of the proteasome. In AD, that was published by Bill Marksbury at UK a couple of years before he died. And because of that, there's going to be a consequent accumulation of aggregated and damaged proteins. That's the quintessential hallmark of pathology of AD, is these aggregated damaged proteins. So how about CRMP2? This is collapsin response-mediated protein 2. This protein is involved in extending one neurite from an adjacent neuron to another to make a synaptic connection. So if CRMP2 is damaged, you would predict that the neuritic length in an Alzheimer's neuron should be shorter than the neurite length in normal brain aging. And you would be right. Paul Coleman showed that in 1989, I think, at the University of Rochester. So again, the proteomics-identified proteins fit the pathology of what you see in AD brain. And how about PIN1? So PIN1's involved in a lot of things. So we have an RO1, have a PPG. My project is on PIN1 in our PPG. And this is about PIN1. So we have a review of the PIN1 in Alzheimer's. So basically, what happens is the WW domain of PIN1 recognizes, so PIN1 is a regulatory protein. I should have said that. It regulates the activity of its targets by modulating the structure of the proline that it's involved in. So it's a cis-trans, it's a prolyl, let's see, what is it? It's a peptidyl prolyl cis-trans isomerase. That's what it is. So the WW domain recognizes a phosphorylated serine or threonine on the N-terminal side of a proline of the target protein. So once that binds, then the catalytic loop loops up. This is a hinge protein, so it loops up, and that catalytic site converts the proline from cis to trans and thereby changes the structure of the target and thereby regulates its activity. And that's what it does normally. Okay, but now in AD, what happens is if the target, for example, is APP, and that's a known target of PIN1, then the protein kinases and the phosphatases are all going to work normally, and you'll get non-amyloidogenic processing of APP, decreased plaques, healthy neurons. But because PIN1 is oxidized, it's possible that you'll have the cis form of this and never can get it back, and that forces APP into amylogenic processing, increase A beta and plaques, and cell death. And likewise with tau, tau is another target of PIN1. So the things like GSK3 beta and CDK5 are known kinases for tau, and PP2A is the known protein phosphatase for tau, and both of those groups are regulated by PIN1. So when PIN1 has these proteins in their trans and keeps it in a trans configuration, then everything is healthy. But when it's oxidized and these targets are in the cis, then they can't be brought back to tau, brought back to the trans, and so you end up with having fibrils of or filaments of hyperphosphorylated tau that are resistant to the protein phosphatases, and PIN1, then tau, hyperphosphorylated tau, falls off microtubules. As a result, you get this dysfunction and tangles. Just think of the consequences of that, right? Mitochondria down by the synapse, no way to get back to the cell body. Therefore, no ATP being made down at the synapse. That's why I believe that the synaptic region of AD, known to be the first part of a neuron to die, is one way is because of PIN1's effect on tau. Okay, so how about gamma snap? So gamma snap is part of the machinery that docks neurotransmitter vesicles to allow them to release their material out into the synaptic cleft for binding to the postsynaptic side. So you would think then, if gamma snap is oxidized, that docking is going to be dysfunctional because it's key to the proper orientation of everything. And as a consequence, you would say neurotransmission is going to be disrupted. And we already know that about cholinergic systems and others in Alzheimer's. So again, this is consistent with this idea. And last example is in the VDAC1. So this would conceivably then lead to the opening of the mitochondrial permeability transition pore, and then cytochrome C and APAF could come out and form the apoptosome and lead to apoptosis. So consistent with that, some years ago, we showed that in AD and MCI, we see elevated pro-caspase 3, which is leading to caspase that could be consistent with this process. Now, if we repeat this entire process over, this time in brains, rapidly obtained brains from amnestic MCI individuals, we come up with exactly the same category of proteins. And it suggests to you, and to me, I hope, I mean to you, I hope, that I already suggest to me, but I hope it suggests to you that oxidative damage to these proteins is not a consequence of the disease. These could be involved in the progression of the disease. This is happening way before dementia happens. This is happening when oxidative stress is present, and only thing happening is memory loss. So that's pretty cool. So if we look at kind of what is common between MCI and late-stage AD and early AD, for example, we see it's enolase, ATP synthase, complex 5, and UCHL1. This is for protein carbonyls. If you look for HNE binding, it's enolase and ATP synthase. And if you look at nitrotyrosine, it's enolase, and depending on what the combinations are. But you see enolases keep showing up. And I mentioned to you how important this molecule is, more than just glycolysis. So let me sort of tie up this idea of progression with the idea of preclinical AD. So these are cognitively normal people. These people have score in the normal range in many mental scores or other instruments of cognition. Now, all of a sudden, they die of a heart attack. They have been followed, by the way, through our normal controls. These are normal controls, we think. They score in their thing. Now they die of a heart attack. They die in a car wreck. And within 2 hours, we have their brain. You look inside and you say, holy smokes, that's an Alzheimer's brain. It's loaded with plaques and tangles. Not as much as a full-blown AD, but a lot of pathology. Now, we never will know because they didn't live. But the implication would be, had they lived long enough, they would have developed Alzheimer's disease. We don't know. So that's a whole area of...
I'm sorry, can I ask a question? But there are cases like that where people are 70 years old.
Yeah.
And you see they're still loaded with A beta and tau, and they do not have Alzheimer's disease. So the implication is not necessarily that they will develop Alzheimer's disease as they age.
It's
right?
true. It's true. There's one other thing I didn't talk about. That's called cognitive reserve. Synaptic reserve. Think about that. You know, did your mother, when you were little, read you books and stories, sing to you, make you do tactile things so that you had to make lots and lots of synapses? And did you stay active intellectually your whole life? Then you have a reserve. You know you're going to lose synapses as you age. Now the question is, how about, do you have enough? The pitch is these people who die with plaques and tangles through their brain still must have had a lot of synapses to stay healthy. One more thing. I submit to you, and I'm not alone in this, that plaques aren't necessarily a bad thing. The bad thing are oligomers. So the brain is saying, oh, my goodness, look at all these oligomers. Let me make these oligomers into a fibril, and let me get these fibrils into a plaque, and they're extraneuronal, right? because there is no bloody way a plaque can enter a bilayer to start the process of lipid peroxidation. You saw our model. Lipid peroxidation has to be the first step. It's clear to us that's happening. And we did the kinetics in this, by the way. I forgot to mention that. We showed A beta causes lipid peroxidation within 10 minutes of exposure. Protein oxidation is 30 minutes. So lipid peroxidation is first thing.
Have you had many people say in the 60s and looked at them this way? And are there people free of this problem? In other words, how many people have you looked at in that decade of life?
Imagine preclinical AD is a rare event to actually have pathology on.
Yes.
Because, they're going to live, right? And then all of a sudden they die of a heart attack.
You were taking people, say, automobile accidents.
No, we don't take people from automobile accidents. What we take are people who've gone through our memory disorder clinic. So we know them because they're normal volunteers. We have the world's largest volunteer to give us their brain when they die, 600 normals.
So you never look broadly at
who would have nobody has the resources for that.
Yeah.
So we are able to look at our memory disorder clinic, and those individuals we know. Now, all of a sudden, they die. They've already agreed to give us their brain. We get it, and we look inside and say, holy smokes, what's going on?
Something to say with regards to what Dr. Butterfield is saying is that this is also has been amply confirmed by the live imaging studies.
That's
so
right.
we don't need to really wait until people have died and nowadays. And so
That's true.
Yeah.
John Morris, for example, at
Exactly.
WashU has now got a very large study of live imaging by MRI mostly, by other
Like I've heard, at least from the UCSF memory clinic, that they get 25 to perhaps even up to 30% of their normal cognitively tested controls that show pathology compared.
Right. At age 85, 50% of the population has Alzheimer's.
At age 85.
Age 85. So that means they had to start somewhere, right? And go on with it. So anyway, so we looked at preclinical AD, and we did, this is not Redox proteomics, but expression proteomics, and I'd just like to point out pin 1. Now, unlike Alzheimer's and MCI, there is an elevation of pin 1 in preclinical AD. We have 10 brains that we can work with at UK, and all of them show this elevation. Now, how do we interpret this? What we suggest is preclinical AD, the brain is saying, holy smokes, look what's coming down the line. And my brain is still elastic enough to be able to respond to this, and so it upregulates a protective gene of PIN1, and thereby hopefully protects itself. But at some point, that battle is lost as more and more free radicals come on, and when you go to MCI, and so forth.
How about the level of oxidative damage in this?
None.
That you can't see it?
We can't see it.
Okay.
Now, when we compare preclinical AD to amnestic MCI, what is the difference? Cognitively normal, no memory loss, memory loss. So if we identify proteins that are involved in that process, maybe we have learned something about memory, or at least the loss of memory. And so when we did that, we published that recently. So here's the oxidative damage. So preclinical AD, no difference than control. Again, really fast autopsy program. But in amnestic MCI, significant elevation of oxidative damage. And when we do the Redox proteomics, the protein that shows up is enolase. So the difference between PCAD, preclinical AD, and amnestic MCI, just looking at those two, they both have roughly the same pathology. And MCI has a bit more than preclinical, but roughly the same. The difference is MCI people are amnestic, and enolase, there it is. And so it's consistent with the idea that preclinical AD is still a relatively protective period in which the brain is plastic enough to be able to fight this problem. But also the importance of the oxidative dysfunction of enolase has in loss of memory. You just saw a specific AIM-1 of our newest R1 that we're going to do on this. Okay, and I told you about this before. Okay. And I won't have time to go over all of this, but basically we can recapitulate the proteomics identification of proteins that we see in AD by the use of A-beta 42 in vivo or in vitro models. So if you want to see that, let me know. So, but I'd like to just quickly tell you about the beagles, because the beagles, you know, they have aging process, and they develop kind of plaques. They don't have tangles, they have plaques, and they're made up of A-beta of the same sequence as humans. So we hypothesized that this aging beagle that's rich in human A-beta, if we gave these, and these are old dogs, these are 12-year-old dogs we start with, and we give them this treatment for 3 years. A high antioxidant diet and a program that makes them make synapses. So the old expression, you know, teach an old dog new tricks. So we basically do that to make them make new synapses. And now you have a 15-year-old dog, and our colleagues at Irvine did the memory tests on these dogs, and they made the same mistakes as a 4-year-old dog. Isn't that amazing? And so they sent us the brains of these 15-year-old dogs compared to dogs treated with just given dog chow. And what did we see? Well, if you have enrichment, environmental enrichment, that means making synapses, and antioxidant diet, you have a marked decrease in oxidative stress. Nothing else alone would do it. This combination would do it. And when you do Redox proteomics, you find these proteins, notice these numbers, the dog that had dog chow would have the value of 100. So anything less means less oxidized. And there you see glutamate dehydrogenase. And I know you know how important that is in the Krebs cycle. So to go back into alpha-ketoglutarate or to go backwards to make more glutamate. So it's less oxidized. GAPDH, we just talked about. Enolase, NFL, GST, and this other protein that involves actin bundling.
So one thing I was saying, so your GAPDH, did you ever see in the nucleus? Because
it does go to the nucleus as a monomer. As a monomer,
right?
Yeah. Not as a tetramer. Yeah, I haven't seen it, but other people have. I haven't looked. I mean, I just haven't looked. But anyway, if you look at all these pieces, what you see is proteins involved in increased energy metabolism, reduced oxidative stress, improved calcium homeostasis and maintenance of LTP, long-term potentiation, and reduced oxidative damage, stabilizing cytoskeleton. All of these processes very likely contribute to the improved cognition these dogs had. Now, can you translate that to people? What if we did? What it would suggest to you, all your life long, what should you do? 3 things. Make sure you eat a high antioxidant diet, which of course includes a glass of red wine every day. And you make sure that you stay intellectually active your whole life. I'm trying to learn some Italian, and I know just enough now to be really dangerous. It's terrible. I'm terrible. But I can order food, at least in Italy. So, but in any case, and I find out which train to get on in Italian. But now the third thing, so you got to do this your whole life. Don't stop ever learning. And the third thing is exercise. Those 3 things made those dogs look like, a 15-year-old dog look like a 4-year-old dog. Yeah?
If you go back a slide, I understand how the diet might, these are the enriched and antioxidant fed. Why would enrichment have an effect on the oxidation?
Oh, that's an excellent question. You know, in other words, how are we able to maintain this? And we don't know how to dissect this out. We don't yet. But for sure, enrichment alone is not enough. Antioxidant diet alone is not enough. You have to do both. And the intersection of those probably comes about from better synaptic function and long-term potentiation. I suspect that's how the learning and memory are improved. But honestly, we don't know. Right now, this is an observation, but and now we're going forward with our statin studies. We've done all kinds of studies of statins on these dogs, and going for there.
How is keep you intellectually active fit here?
Well, if you're intellectually active, you're always making synapses. And that's my point. You have to learn new things in order to keep making synapses, in order to keep your brain active enough. Because as you get older, you're going to lose synapses. What are some of the major risk factors for AD? Age. What's the second biggest risk factor besides traumatic brain injury? You think about all those poor fellows and women in Iraq and Afghanistan. What is the second, third risk factor? Illiteracy. So if you start with a low level of synaptic load, you are in deep doo-doo when you get older. Yeah.
I was curious about the antioxidant diet. Have you seen a dose response with that? I mean, have you looked at...
This is all worked out by the Irvine folks, so I don't know all that. But I didn't show the diet because it's just for time, but the diet had grape, carrots, lipoic acid. That's an interesting thing. I always like to ask graduate students if they know the major complexes that are supported by lipoic acid in their function. Anyway, let me go on. So I don't know if I have time. How many minutes do I have left? I can go on? Because I do want to get to the mitochondria in living people, because I want to get to that for sure. So this is a study that was done with the help of Veronica and Dale Bredesen when they were at Buck Research Institute. So basically what happens is you take the J20 mouse, which is a Swedish-Indiana double mutant, human double mutant transgenic mouse of APP. And we asked, so I gave a seminar out there and Dale looked at that and said, oh, you know, we had long ago shown the methionine residue of A beta 42 was essential for its oxidative stress. So he said, what if we build you a mouse in which that methionine is missing? What do you think we might find? So I can't believe how hard they worked to make that third transgene in here, but they did, and they succeeded, and they built a mouse now with a third mutation, which is methionine 631, which corresponds to residue 35 of A beta, and they converted it to leucine. And now, if our idea is right, that oxidative stress depends on the presence of this methionine, then here we have the first in vivo mammalian study to show that. We've already shown it in worms. If you take human A beta 42 and express it in C. elegans, you get oxidative stress. If you take the methionine and convert it to cysteine, do the same experiment, you get the same deposition. There's no difference in deposition, but there's no oxidative stress. So that methionine is really important when it comes to oxidative stress. So here's the sequence of A beta. Here's this S. So we made the sulfur. This is in cell culture. We made this sulfur into a CH2. This is called noroleucine. And when you look at that, we saw no changes in oxidative stress. And I mentioned the C. elegans. There's one other study here. The alpha helix secondary structure is important. So if you remember in the bilayer, the model is A beta is in the bilayer. Every peptide and protein that you know of, with some exceptions, there are some, in the bilayer adopts the secondary structure of helix. The helix has an I+4 rule. Every fourth amino acid interacts with each other. NMR shows very clearly the carbonyl of isoleucine at 31 is within a van der Waals distance of the sulfur atom of Met35. Oxygen is more electronegative than sulfur. Can you see this in your head? How many see the sulfur atom here? You see it? There it is. My thumbnail with 2 electrons hooked on to the sulfur nucleus, valence electrons. Left hand, sorry. My left hand is oxygen of ILU31. It's very close to that electron pair. Which is more electronegative, O or S? Oh, so electron density is going to move from this sulfur towards that oxygen, meaning that lone pair on the sulfur is going to be held less tightly. So any oxidant can come in and make a one electron oxidation of that lone pair and bring that electron over here and to the oxidant, not to the oxygen, to the oxidant, and leave a a sulfur-centered radical, S+ dot. It's called sulfuranyl radical on the met. What will that do? I wish I had shown all these slides. S+ dot can take an allylic hydrogen off the acyl chain of the phospholipid instantly and make a C-centered radical on the acyl chain of the lipid and a acid SH+ on the sulfur of met. Guess what the pKa is of that acid? Answer, -5. That means any base instantly takes the H+ off. And what do you come back with? Met. What do you call a reaction in which what you start with is what you end up with? You call that a catalytic reaction. So the A beta is acting like a catalyst, and that's why you don't have to have very much of this happen to greatly amplify the effect, because you have an infinite amount of acyl chains with unsaturated chains, all of which have allylic hydrogens, which are highly labile and can be pulled off, and you just keep that chain reaction going and going and going, and you get an enormous elevation of oxidative stress. So we published all this mechanism in the Journal of American Chemical Society. It was a pretty tough journal to get into, and in 2001. So I think if you want to know that a little bit more, you can see that. And then also we did one other experiment. If we take the residue 37 as a gly, and suppose we put aspartic acid in it, now you still have a helical structure if the molecule is in the bilayer. But because it has aspartic acid, it's as though the peptide is down here. Here's the surface of the membrane, so here's the A beta. Now, let's say where this pointer button is where the aspartic acid is. That's a negative charge. It will pop up. So it can make all the free radicals at once. It has no substrate with which to react because it's outside the bilayer. And when you do that, you find absolutely no oxidative stress. So that's all consistent with this finding. And so look in JAX if you want to see that paper. So Veronica and Dale built us this mouse, and the third mutation. So the hypothesis would be the brain from this mouse would show significantly decreased in vivo oxidative stress, and it'd be the first time that's ever been demonstrated in mammalian system. And so that's what we did. So the MET would be gone because it's replaced by LU. And so here's what you find. So here's the non-TG control. Take that as 100%. And here's the J20 mouse, as you would expect. There's a ton of A beta being produced. It has the MET in it. You're going to get oxidative stress out the wazoo. And here's the non-TG M631L, which corresponds to residue 35 of A beta. And now, as soon as you make this substitution, look at that. There is absolutely no oxidative stress there. So that is telling you that methionine is absolutely critical to the oxidative stress of A beta 42. Isn't that really nice? And these are by different mechanisms. You see the same thing. And then out at the Buck, these folks measured A beta 42 and A beta 40, and the ratio, no different. So this stuff is still dumping all the material. However, the form is different. And that's what's kind of interesting. If you look at in the one that has the methionine missing, the A beta is in little punctate pieces, whereas in the J20 mouse, Swedish Indiana double mutant, you get the usual kind of deposits. Well, if you again translate that back to AD, it might suggest that this methionine is really important in plaque formation as well. And actually, if you know peptide chemistry, you'd believe that, because Sam somebody, University of Wisconsin Chemistry Department, he built a cyclic peptide of 18 amino acids, and he took a cyclovoltammetry instrument and converted the met to methionine sulfoxide. And as soon as he did, the thing went to beta sheet. Then he turned back the current voltage and went back the other way and made it into a random coil. And then he did it back and forth. He could just do that over and over. So, and we know plaque of A beta is beta sheet. And about 50% of plaque has methionine as methionine sulfoxide of A beta. We know that from a 1994 paper, Naslund et al. In PNAS. So the pathology is interesting. So it suggests that in this study, at least, A beta shows that this study shows A beta is a major source of in vivo oxidative stress in both AD and MCI. The single met is important, and maybe for plaque formation as well. Okay, so I showed now, here's the proteomics of these mice. This paper is recently accepted in, recently published in ARS as well. And I don't know if I want to go over all this, but basically the bottom line to this study is when the MET is missing, the oxidative stress in the brain is much less. There is no need for upregulation or compensatory responses to the oxidative stress. And so what you see are about the normal levels compared to the non-TG mouse, and much less, usually, than the J20 mouse, where the oxidative stress is there, and so the brain has to respond to these oxidative stress to try to overcome it. Yes, Arlen?
Do these mice show loss of cognition?
Well, it's interesting. It's interesting. We had one measure that the folks at the Buck did, and that was the water maze. They made sure that there wasn't any blindness issues or muscle differences or so forth. And by the water maze, we really could not see an improvement in cognition. So initially, I was really bummed by that. But then Dale and I put our heads together, and maybe, Veronica, you helped in this too, but I didn't talk to you, so I don't know. But with Dale, for sure. Remember about APP. APP is processed by beta secretase, and what is removed when that happens? When APP is processed by beta secretase, what's removed is secreted APP beta. That's a highly toxic peptide that binds to death receptors and leads to neuronal death. Did you all know that? There's nice studies on that in Nature, I think it was, recently. And then Dale has shown that APP is processed such that the cytoplasmic component of APP produces around, forgotten the number, but at one residue, a peptide that, if it goes to the nucleus, can induce apoptotic death and cause that. So the point is, we have stopped the oxidative stress, which is clearly due to A beta, but we haven't stopped the production of toxic peptides.